Good day, students. This recording is aimed for the subject Food Analysis 2, and in this section we're going to be discussing the theory related to methods that are used to analyze for minerals. And in this video we're going to place greater emphasis on the determination of sodium chloride using the Volhard method, determination of sulfur dioxide, and then the determination of the bar value, or what we call the titratable acidity. Just as a reminder that the work that has actually been covered in this presentation is merely a summary of the book Food Analysis by Suzanne Nielsen. So please do consult this textbook for in-depth understanding. The textbook is actually found on the library portal, and you can actually download that and keep it for yourself, because we use it for almost everything. Okay, so in terms of our minerals, the nicest thing about this chapter is that we've already started analyzing for the minerals when we analyze for the ash content. And if you can still go back to the procedure there, that's where you take a sample, you char it, and after that you continue the charring in the muffle furnace for 5 hours. So in that process, what actually happens is that the organic matter that makes up the food matrix then is degraded, is burnt away, and then what's left is the inorganic matter, which then is known as the crude ash or the crude minerals. We call it crude because we just say it's total minerals, but we don't actually know what's in there. However, the methods that we are going to talk about, those three methods that I mentioned earlier on, your sodium chloride, sulfur dioxide, and titratable acidity, goes a step further and specifies which minerals are actually in the food product, and it also quantifies as much as to how much of that mineral is actually found in there. So it's not just as crude, it goes down to specifying the amount and the type of mineral. So of the specific mineral determination, we have three main ways, or five main methods. The other two methods are discontinued because they are not user friendly, so that's why I said there's three. So we have the titrimetric, colorimetric, and then the ion selective electrodes. And the reason why these are still used today is because the chemicals that are involved in these methods, or that are used for these methods, are still available, easily available, and that also goes for the equipment that's used for it. Unlike the two, the gravimetric and the redox reactions, they are quite difficult to, or complex in terms of analysis, and sometimes getting the chemicals that are used for those might also be difficult, and they are not so used anymore, applied in food science and technology. So that is where we focus on the three main ones. And then the other one is that the fact that those three are the easiest, most lab technicians, even boiling down to a new person in the lab, will find it easy to analyze compared to the other ones. And then what happens is that since these are the mostly used methods, so what they do is that they are then used as a basis for modern kits, you know, the fast kits where you can just analyze for salt in a few minutes. That would be due to the principles involved in those three main methods that I mentioned there. Okay, so the importance of your minerals in the diet. I mean, we cannot just go about saying, okay, we want to analyze minerals. So what about minerals? The first thing is, first, in terms of us food science and technology people, we care mostly about food in terms of the functionality. And then nutrition also plays a role, but we are more of the functions of that in terms of food processing. So in terms of the importance of minerals in the diet, your minerals in the diet serve obviously a major purpose, and depending on that major purpose, they can actually be divided into different categories. We have what we call your macro minerals or macro elements, and those are actually required in higher quantities, and those higher quantities are something like more than 100 milligrams in the body per body weight of a person. And those are called macros, so they are required in higher quantities. So you have your calcium, your potassium, your sodium, and your magnesium. And then from there, you also have your trace elements. Trace elements, those are required in smaller quantities compared to the ones that I just mentioned now. You have your iron, you have your copper, and you have your selenium. And then we also have the ultra trace minerals like your nickel and vanadium. Those are required in even smaller quantities compared to the other two. And then the ones that we actually do not want in food, those are called the toxic minerals, like your lead and your mercury. So those will be detrimental to a human health. That is why we then do analysis to be able to quantify or identify if those are in there. And then, as we already know, these minerals have an important role in the diet. I mean, if you talk about your calcium, we already know that your calcium will be beneficial in terms of your bones and your teeth. And then you talk about your sodium, which then obviously going to balance out the salt osmosis that's taking place in your body. You have your iron that is there for your red blood cells and so on. So each and every of these minerals, they play a major role, and that is why they have to be in the diet. And when being in the diet, we have to make sure that we are not deficient of those. So that is where you as a food technologist come in place, whereby you analyze and you tell us how much of it is in there. Then we can actually make an informed decision that, okay, there's enough of this, let me consume it. It's going to do 1, 2, and 3 in my body in terms of the functionality. And then very important also why we analyze is that it is actually mandatory for us to analyze for things like your sodium chloride. And obviously this is due to the regulation. Like the other very important one is R146, which is actually a regulation that relates to the labeling and advertising of food stuff. So it's required. It's not a, okay, I can do it if I want to. It's mandatory. It has to be done because of the importance of your sodium in terms of health in food products. And then also other minerals, you are only required to analyze for them if you want to make a claim to say high in calcium because it builds stronger bones and teeth and whatsoever. So in that way, that's when you will have to quantify and tell us how much it is so that we can believe that it will definitely be enough to make your bones strong whatsoever. Okay, so then as I had already mentioned that in terms of us food technologists, we are not nutritionists. As much as obviously we work with food, but we focus more on processing. So what is the importance of minerals in food processing for us? So as I already mentioned that it will serve both your nutritional and functional property in food, but we are more interested, not more interested, but we know more about the functional side than the nutritional. Okay, so in the nutritional and functional side, we still need to control those because like I already mentioned that if you need 100 milligrams, but now all of a sudden you have 500 milligrams, we all know that too much of a good thing is also bad, isn't it? So it then becomes toxic. So that is why there should be some form of control. Okay, so as we already know that certain foods are actually naturally high in certain minerals and some are not. And based on the type of processing that we do, I mean, we heat. Heating can either be your pasteurization, sterilization. We also dry at high temperatures in the spray dryer and so on. So what happens is that some of these minerals can also be lost during processing. So the other important side of analysis is to see if there was any loss due to processing. So that's another important aspect of mineral analysis. So making an example, in terms of milk, milk contains high levels of calcium. However, when cottage cheese is manufactured using the direct acid method, what happens is that calcium becomes leached into the whey, and in that way it's actually lost. So the cottage cheese will then have lesser, because remember the calcium is found in the protein there and then in the protein casein. So it leaches out into the whey protein, so that the resulting cottage cheese then has less of your calcium. That will be the first example. The second example will be when you are having grains like let's say your wheat. You know, when you have whole wheat and processed wheat, wheat for white bread and wheat for brown bread, and we already know that when the processing of removing of the bran, what happens is that most of those minerals will be concentrated in the bran to make your white flour. So it means then your white flour will contain less amount of your minerals. So the flour that is resultant from that process will then have lesser minerals. So therefore we lose those. So what usually happens is that we can actually replace some of that, and that is known as enrichment, whereby we already knew that this as a raw material, it used to contain so much iron, and that iron was actually lost during refining or whatever process. Then we can add that iron back so that we enrich it again. However, fortification on the other side is something that is different. It is also similar to enrichment. You are adding minerals, but you are not adding what was lost. It's either it was lost, or it was not lost, or you never had it at all. So in South Africa, it is actually mandatory for you to fortify things like your maize, your bread, your wheat or your flour, and then also your salt with iodine. So why? Because we don't want any deficiency. So most of the times if you look at those products, they would have a small sign like this. Especially I see this mostly in the bread. Okay, so minerals are very important in food processing, and we also don't want to lose them. And then we also know that sometimes minerals can be very much higher than what it is expected, especially your sodium chloride, your calcium and phosphorus that are found in different food products. So nutritionally, we already know that in terms of the different types of minerals, we know that nutritionally, your salt is then fortified with iodine, and that is obviously to prevent the formation of goiter. Goiter is that big plump that is normally forming here on the neck. So it becomes a nutritional problem, because if you don't have iodine in the salt, then obviously goiter forms. So that is actually a nutrition related problem. But as we come to the side of food science and technology, where we talk about processing, we focus on functional properties. I mean, we all know that meat that is not salted does not taste nice. There has to be salt. So why? Because salt adds to flavor. And then secondly, salt acts as a preservative, because remember, microorganisms require water. So when you add salt, what happens is that the salt actually takes up all the water, and then what happens is that when the microorganisms grow in there, that salt can still draw water out of the cells of the microorganisms, and in that way it can actually kill the microorganism. I mean, think about the salting of snoek. And then it also helps in terms of increasing the ionic strength. Like when you are making brine, you guys know brine is a salt solution. So the more salt you add, the more you increase the ionic strength. So therefore, you also bring about that preservative effect of brine. And then another very important aspect that we're going to cover in the chapter proteins, whereby your salt is actually used to solubilize protein. And what it means is that the salt is actually used to ensure that the protein binds with more water, and in that way it's a functional property. It brings about the texture of the product. And then also you also have your minerals that promote the gelations of proteins and gums. This will be your calcium that is actually used in the making of jams and stuff, and in that way it helps to make the jams gel, if you put it that way. And then very important in terms, it also helps in terms of water holding capacity, and this phenomenon is very important in meats, whereby they add phosphates and brine to be able to hold the water capacity, and then in that way the product is nice and moist. So those are what we are more interested in, or something that we study about more than the nutritional side of the importance of your minerals in food. And then another very important thing, the reason why we also analyze for these minerals, is to also determine the water quality. You know, you've probably heard when they say hard water. Okay, so what happens is that some water will obviously contain some of these micro and macro minerals, and then that can become a problem. So that is why in the lab we only use distilled water, because water contains minerals. Just look, when you have a kettle at home for boiling your water, at the bottom, if you keep on boiling, you will see that it turns like, it becomes a little bit like powdery, and that is due to the fact that you are boiling the water and then some of the steam is evaporating, and then that powder of minerals clogs at the bottom of the kettle. I don't know how. Yeah, maybe you understand this example. So that becomes a problem in the food industry, because when you have high minerals in water, it forms clouds in beverages, and you don't want a cloudy beverage, because a cloudy beverage looks like a beverage that contains a growth of microorganisms. And then secondly, if your water contains high nitrates and you are using that water in processed meat, what happens is that the meat will look pink. It will be as if it's cured when it's not cured. So that is why it is also important to be using distilled water, or water that has been processed to remove those minerals, so that it does not have those negative impact on the food product. So that is why it's important. So you can already see the emphasis. Okay, so now in terms of, we've spoken about the importance of minerals and obviously the methods, but now we have to talk about the things to consider, the nature of the analysis. So when we are analyzing, how do we actually go about it? What will be the first things that you do? Okay, so the basic analysis is, we actually are looking for minerals that are found in food, but you cannot pinpoint where the mineral is in the food. Remember, food is a matrix. It contains all things that are bonded together. So in that way, it means that if we are looking for that specific small mineral, we have to separate it from the matrix. And after separating it on the side, then we can measure how much it is, because whilst it's actually attached to other components in the food, that becomes an interference. Okay, so what will be those basic analysis? Like, you separate and then you measure. So the first example, for you to be able to separate the analyte or the mineral that you're looking for, we have what we call the complexing, or what we call precipitation titrations. So what is it done? It's actually used to actually separate the components that you're looking for, and then that is just used by precipitating it out of the big matrix. You know, it's like you just extract it, but when you extract it, for you to be able to obtain it, it will be found in a precipitate. Okay, so then you can actually get that specific analyte that you're looking for. So what happens in those type of reaction, the volume of the titrant will then be equivalent to the mass of the mineral. So, and that is obviously based on the stoichiometric relationship as to this one reacting in a 1:1 or 2:3 or whatsoever. Okay, and then, so besides you precipitating the analyte out, number two, what you can do is you can separate it, because precipitation is also separation, but it's separation of the analyte, but it's still within the system, but it's just that it's precipitated to the bottom. But secondly, what you can do is you can separate the minerals, and you can actually do that by ashing or acid extraction. Like, remember we mentioned ashing? So we just did ashing, and then we just said, okay, we're just looking for the crude minerals. But in other analysis, you can go further. You can take that ash and then continue with it and do other tests. Okay, so what happens is that after you have ashed or you had done an acid extraction, you can then do specific measurements to determine the content of a specific mineral, because remember, now you have gotten rid of all the organic matter by burning and charring, and then from there you can then identify the specific mineral within that ash that is formed, And then you can use colorimetry, you can use your ion selective electrodes, atomic absorption spectroscopy. Those are one of the latest equipments they use out there. We don't have any of that in our department, but other departments in the applied sciences have that. And then those ones will require that you need to first do the etching. And then the third one is you would use a standard curve. I think you guys have done standard curve, whereby you probably did a standard curve of potassium manganate in chemistry 1. So using a standard curve to determine the concentration, what they do is that, let's say you're looking for a concentration of iodine within a sample. So what you're going to do is you're going to prepare different concentrations of your iodine, and then you plot that in a graph based on the concentration versus the absorbance on a spectrophotometer. Then you have your nice straight line, and then when you analyze your food product, you can be able to extrapolate the actual content of the iodine in the product. So that will be the use of the standard curve. Okay, so that will be what you do. Remember, we said you separate and then you analyze. But obviously, before you do everything else, there must be sample prep, because you might be analyzing your minerals from, let's say, a meat product, from fruit, or from fruits, or from a powder in a silo, in a big silo somewhere in a warehouse. So you still need to apply those sampling technique that Mrs. Minnaar taught you. How do you go by identifying how much of the samples you take? After that, then taking the representative sample, then what do? Then you have to do the grinding and the chopping and doing whatsoever, making sure that you reduce the particle size, because remember we mentioned that those analytes or minerals are found within the matrix of the food, embedded inside. So for you to be able to do the extraction, you must make sure that you reduce the particle size. But very important is that whilst you are doing that sample preparation and whatsoever, you prevent any contamination, because you don't want to be analyzing for salt or for iodine or for aluminium, and then you contaminated that the sample that you are using to collect is also aluminium. It's an aluminium dish or whatsoever, but you are also looking for aluminium in that product. So the shavings from the aluminium dish might then contaminate your sample. So it's very important that you note the things that might contaminate your product. Okay. And then also in sample preparation, obviously the purity of your chemicals is very important. And then also the using of the blank reagent. I'll talk about that just now. Okay, so then from there, after you do that, let's say when you are measuring a blank, it's when you have a procedure whereby you have all the chemicals. You are reacting sodium chloride, you're reacting your sample with your sodium nitrate, however, or your sodium nitrate, your nitric acid, everything. But when we talk about the blank sample or the blank preparation, that's whereby you talk about everything else without the sample itself. That's why it's called the blank, and it's normally used to calculate the difference between the sample and then the titration. Okay, so another third thing that you have to worry about will then be the interferences, because of the different type of tests that are used. It might be affected by certain environmental conditions like the pH, like your one of the methods that is used for salt determination. The Mohs way of salt determination requires the pH of the product to be between 6.5 and 9. So it means if the pH is out of that range, that method is quite useless, or the procedure will not be accurate enough. The sample matrix can also be a problem, whereby you are not being able to grind it so that you can reduce it to smaller particles. The temperature, maybe the product might be very hot or very cold. And then analytical conditions, and then reagents. Reagents, we're talking about the purity. Maybe you're using a non-pure reagent. So that will then be your interference. So how do we go about preventing those interference? Because, I mean, if there's interference, it means that the analysis cannot be done, but the analysis has to be done. So those interferences can be overcome by either boiling to remove your carbonates, because that will then interfere, and then also using of standard curves generated from the food matrix, and in that way you can get the actual concentration without any interferences of those chemicals in the food. Okay, so I'm gonna stop here and then I'll talk about the methods.